some photos of all the different stages of meiosis. You can use this as a reference and information guide to help you guide yourselves through the process of meiosis. I'll be coming down to each group one at a time to see where you guys are, where you're struggling with, where you are doing well, and also if you guys need help if you need it. So, in just a moment, I'm going to come around, I'm going to pass you guys the worksheets in each of your groups. What you should be doing right now is modeling two homologous pairs of chromosomes. Guys? And as I pass it out, you should have already decided who is going to do what in your team. Are you recording that?
Yeah.
Alright. So, here is the questions packet. Figure out who's responsible for that. You turn in hard. All right. Here is your modeling. It's a good drawer, of course. Alright. So, here is the modeling packet for you guys. Thank you. Modeling packet. Modeling packet. Is the modeling packet? Alright guys, so remember, you want to go ahead and build your chromosomes so that you've got these sister chromatids as one chromosome. And then here is another one that would be homologous to each other. You can design it any way you like, but remember, they've got to be identical exactly when they're sister chromatids, and they've got to be similar. The same number of beads, the number of genes all the way along the way. Make sense? So here's one homologous pair, and I'm pulling out another one right now. Buried in my pocket.
How much do you have to make? We have to make 8?
So you'll have to make 8 total different groups, but you'll be basically making 8 homologous chromosomes. You'll have 2 homologous pairs. Yes. Yeah, that one's for the drawing. And I bet you're probably going to be awesome at that. So remember to go through the different phases. Okay? Alright. Alright guys, so I'll come back and check on you in just a moment, alright?
Thank you. So we joined here, right?
Yeah, that's where you're going to be drawing, right inside there. Now you've got 2 places to draw. This is just one right here. This is the main one right here. You have to go through all of the phases, each one of them. Now, obviously, like we did last time for my toes, you're not going to trace that and fit right in there. It won't fit, will it? So just, do your best to do your representation.
Someone destroyed my chromosome.
Oh, easy to fix.
Yeah, I can do anything.
I left it on the table.
See, I gotta fix my chromosome.
They all have to
There we go.
How did you destroy it?
There we are. But you see how you can make any chromosome you want. But remember, when they're homologous, they have to have the same number of genes. It's like how these are homologous. Now, Isaac, could I make these become homologous? No, why not?
Because the tips are specific.
Well, don't worry about what color the tips are. It has to do with the size of the chromosome. Are these the same size?
No, because they're missing two.
Yeah, Exactly. All right, excellent. All right, keep up with your work. I'll check you guys a little bit.
Thank you.
All right, guys, so here's some chromosomes that I built. Notice how these ones here are identical to each other because they're sister chromatids. Is this identical to this one? No, they're not identical. But notice how these ones have the same length. They might not be the exact same gene per gene, right? This one could say, hey, make blue eyes. This one says make brown eyes. But because these ones right here have the same length, they might necessarily code for the same thing. They can be homologous pairs. Here's one that will make a tetrad. You guys need to build 2 of them for you. Sound good? You can make it exactly any way you want to, but this is what you want to end up with. Something like this. Any questions, Daisy? Ask me how we doing. You doing fine? Good. All right. Any questions for me before I go? Good. All right. I'll come check you guys a little bit.
Using the 7 pop beads of the same color pop beads for each. Students in Group B will assemble a similar set.
All right. So you're doing a great job of reading the directions. Let me save you some time though. So let me help you figure out what we need to do. So who's our modeler here? Daniel. All right. So Daniel and Hoppy the leader, you are going to go and build your homologous chromosomes. Now to do that, you have to start off with 2 sister chromatids. You see how these right here are genetically identical? So because of that, they're identical, they're sister chromatids. Now this right here is another chromosome. Now, is this the same size as this one? It is. So these could be homologous chromosomes. These could be the gene here that codes for black hair. This could be the gene that codes for brown hair. One from mom and one from dad. They're different, but because they're the same size, they're what we would call homologous chromosomes. Right? Here's another group right here that would also be a great example of homologous. Now, could these here be homologous to each other? No. Why not, Jaquan? They're not even, exactly. So you're going to create the same amount as I've done here of homologous chromosomes. And you're going to form them so they can make tetrads like that. Alright? Now, after you've done this, you're going to then go through all the different stages of meiosis, moving your chromosomes where they will until you get all the way to our product of the end of meiosis.
Yes, sir. So we're making all of them.
You can make it any design you want. Daniel's already doing a great job of designing his chromosomes. Alright. Now, what you're doing? Okay, so maybe you can go and either help Daniel do some remodeling, or maybe help Annabelle over there with some of the questions. That sound good? I'm going to keep those though. Thank you. Alright guys, how we doing? Ooh, really good start. I see you've already got your very first homologous chromosomes together. And why are they homologous? How do I know they're homologous? Not identical. Oh, look at this one. Well, wait a minute. This is interesting. Could these be sister chromatids? No, why not? They're different colors. They're not genetically identical. Yeah. You know what you could do? Oh, I think you're coming with a good solution right now. Brilliant. Brilliant. Yeah. So if you do that, then they would be genetically identical, wouldn't they?
Yeah.
So you just solved the problem of figuring out how to change it. Excellent job, Shemaya. Good. So now you make just another homologous chromosome that could match this. Will that have to be the same as this? No, but what will have to be the same between the 2 homologous chromosomes? Yep, exactly. Same size. In this case, 10. Beautiful. All right, get working on that. Good job. Nice coloring, by the way. Yep. Any other question?
Should we drop that? It doesn't really show you what.
It's a great question. Yeah, so when you see telophase 1, basically telophase 1 is right before you get to this stage.
So it's just split.
Yeah, exactly. So it's now split, and really, we're looking at the end of phase 1. And that happens to be pretty much the start of meiosis 2. Yeah. So you could draw that, the prophase 2, and that would work just fine for that. You see how that works? Good. Because is there really an interphase at the end of prophase, or at the end of telophase 1? No. Just right into it. Good.
What colors did I use? Don't worry about it.
Alright. How are we doing here?
Keep the prophase wrong. You're supposed to get tetra. Wait, those are you didn't get any tetra.
Hold up. So far, you're doing okay, but yeah. This would be really awesome for meiosis 2. But we're actually doing meiosis 1.
God.
I was thinking meiosis.
That's okay.
I'm so smart.
That's okay.
Then what you do?
So really, what's really nice about this is that what you could actually do is just add more, right? So you don't have to erase anything. You just add your other chromosomes.
Oh, so it doesn't fit.
This is the example of sister chromatids right here. And to make it so that you make a tetrad, we just gotta just push them together.
Okay.
That sound good? Cool. So I'll tell you what, if you go and draw an arrow showing this should be coming together, then I'll accept that for full credit. Sound good?
Could I just draw more on the outside? Or is it okay?
You can just draw and push them together, that'd be fine. Remember, you got colored pencils. That can be used to go and help show that which ones are sister chromatids and which one are homologous. Alright, so show that to me with the colored pencils.
I'll cut it later. I'll cut it later.
No, you're not.
Hey, Christy.
Let me borrow your guys' desk real quick.
How do we draw the picture?
I'll show you in just a moment. So first, I gotta get your attention really quick. Eyes on me. So remember, I told you guys inside the classroom that you can't just take your chromosomes and just throw them right smack on this page and trace around them. Will they fit inside this picture? No. Oh. So you would have to improvise. Instead, what we're going to do is that we're going to imagine our entire lab station is one giant cell. And we're going to use our chromosomes as a model as we go through meiosis 1 and 2. To do that, pretend that your chromosomes are right smack in the middle. Throw it off, please. And then you're going to go through each of the phases. First forming your tetrads, pulling them apart so that the homologous pairs go to each side, and then repeating it through meiosis 2 where the sister chromatids separate. Does that make sense? So just model it with your desk, and then go ahead and take that image that you see and place it over your pages for your worksheets. Lucky to answer your question? So you can go ahead and just go and draw a smaller picture of this when they go on. Let me help you with our document right there. Anytime. So, what are your teammates? So they're not technically. Remember that, like when we learned about this in prophase? Are these crossing each other? Are they crossed right now? No. They're just stuck to each other. They look like a little bit, right? So really, this thing could look like this, and it could be like that. We just draw that and model off of it. Exactly. Yeah, exactly. But you're not going to trace it. You're just going to kind of draw it smaller. That'd be a lot easier, wouldn't it? Is that so? How you doing there? Good. All right. And Juan, Mr. Leader here. How's our team doing? We're doing good. Is everybody working on something? So
Just waiting for you to draw that so we can go to the next one.
Oh yeah, he was doing the mom.
you know, you are welcome to go and separate the pages if you want. I have a stapler over there, so you can go ahead and staple them back together. What I'm saying is that because it's due tomorrow, you might want to maximize the time you have here because we only have 15 minutes left of class. So, as a leader, you decide whose time is spent doing what. All right, leader? All right, I'll come back and check on you guys a little bit. How are we doing here? Good? I want to get started on doing some of the drawings. I'm not sure whose job that is. Is that good? Yes. All right, you might want to get started on that. Our model builder here did a pretty good job making us chromosomes. Good work. If you're finished there, maybe you can go ahead and help out with some of the questions or work on a worksheet. Who's our leader? Daisy? So you can help decide how other team members can be effective and helpful. Sound good? All right.
We've already reached how much we got left.
Okay, that's fine.
So, you want me to keep it?
Nah, stop it.
Alright.
Okay.
Yep.